Okay, let's start our meeting. Prof. Gelvin, we'd like to welcome our participants. Already 51 participants join us. Welcome all participants. Welcome our lecturers. I see Pak Wang and maybe others. Welcome in our Zoom meeting. Thank you, Prof. Gelvin, for your time to share your knowledge and experiences with us today. This meeting will lead by Professor Darda Efendi, our professor at Plant Biotechnology Division, Department of Agronomy and Horticulture, IPB University. Prof. Gelvin, IPB University is the biggest university in Indonesia for agriculture. So we do both on basic research because we have also biology department, and also we work on applied research on agriculture. So mostly in our department, we work on crop plants, horticultures, also on medicinal herbs. This meeting will lead by Professor Darda Efendi. We start with Basmala. I invite Prof. Darda to lead this session. Please, Pak Darda, time is yours.
Okay, thank you, Bu Dewi, and good evening, Prof. Gelvin, and good morning to everybody in the side of Indonesia. And I will introduce Prof. Gelvin's, wait a moment. Sorry, wait. What is it? Okay, we welcome Professor Stanton Bruce Gelvin. I hope I can pronounce your name so right.
That's very good. Yes.
Okay. Professor Gelvin is from Purdue University, Lafayette, Indiana. And it's some background of Prof. Gelvin, BA from Columbia University, New York, and major in biology, and then Master of Philosophy from Yale University, New Haven, Connecticut, Department of Molecular Biophysics and Biochemistry, with advisor Dr. William C. Summers, and PhD in 1977 from University of California, San Diego, La Jolla, California, Department of Biology, with advisor Dr. Stephen H. Howell, and postdoctoral research associate University of California in La Jolla, and then California laboratory with Dr. Stevens. And research interest is molecular biology of plant systems, crop productivity improvement by molecular techniques, especially by genetic engineering, crown gall tumorigenesis, and plant microbe interactions. And academic position in assistant professor in 1981, associate professors, and then full professor 1991 to 2008 at Purdue University, and then gets Edwin Amberger Distinguished Professor of Biological Sciences in 2008 until now, and adjunct professor in 2004-2015 in Institute of Botany, Academy Sinica, Taipei, Taiwan, and also adjunct professor 2009-2010 at Zhejiang University, Hangzhou, China. And related work experience, undergraduate research, Columbia University, New York, New York, laboratories of Drs. David Zipser and Ronald Sederoff, and laboratory technician at Yale University, New Haven, Connecticut, 1973. And also working at laboratory of Dr. Yale Nemerson. And lots of awards also, the Mondrian Walter Winsor Cancer Fund Postdoctoral Fellowship, Presidential Young Investigator Award, Purdue University Herbert Newby McCoy Award, Loomis Lecturer, Iowa State University, Fellow of American Academy of Microbiology, and then Fellow of American Association for Advancements of Science, 2010, Purdue University Bindley Bioscience Center Fellow, Japan Society for Promotion of Science for short-term fellowship, and also for long-term fellowship. And then also have as editors, association editor, senior editor, editor-in-chief, association editors, co-topics editor, specifically for Agrobacterium biology, and associates editor for Frontiers in Plant Science. And this also membership of USDA Competitive Grant Program, Plant Pathology, Plant Genome, Corporation for Plant Biotechnology Research, and SF Cellular Process Panels, and also USDA IPAF's co-directors, and advisory committees for International Advisory Committee for Institute of Plant and Microbiology, Academia Sinica Taiwan, Genome Engineering Improvement for Useful Plants or Sustainable Agriculture or GENIUS, and Sweet Tree Incorporation. And lots of membership other organizations, and then publications. Professor Gelvin total says 197 since 1976. Have 13 patents with a high index 64 since 76. And this is some patents of Prof. Gelvin: transcription in plants and bacteria, open enhancement or virulence genes inductions, chimeric regulatory region and gene cassettes for expression of genes in plants, enhanced plant cell transformation by addition of host genes involved in Tdna integrations, a method for delivery of proteins to plant cells, methods and composition for enhanced plant cell transformation, enhanced plant cell transformation by addition of host genes involved in Tdna integration, and others, lots of, mostly on Agrobacterium's roles in agriculture, especially in genetic engineering. And lots of publications, just see maybe too many to say one by one. So based on his experience, research, publications, Prof. Gelvin will teach us about the basics of Agrobacterium roles in genetic engineering, and maybe also in delivery system for genome editing in the last part of the talk. So time is yours, Prof. Gelvin. Good luck.
Okay. Well, thank you very much, Professor Efendi. And I also want to thank Dr. Dewi Sukma for inviting me to give this talk. And I'm going to see, first of all, people could hear me okay? Is it okay?
Yeah. Yep.
Okay. So okay, can you see my screen now?
Yeah, sure.
Yes, yes.
Okay. And I'm trying to remember on Zoom how I get the pointer. Here it is. I think. Yes. Okay, very good. Okay, so again, thank you for inviting me to give this lecture. I've unfortunately never been to Indonesia. I hope someday I will. And I'd just like to talk to you about a background for understanding and manipulating the process of Agrobacterium Tdna integration into the plant genome. I'm going to start with a general introduction about Agrobacterium, and then I'm going to talk about some of our own research work on Tdna integration. And I'm going to end with a discussion of some research work that's ongoing right now about novel ways of delivering genome editing reagents into plants by manipulating how Agrobacterium infects plants. Again, if anybody has any questions during the talk, please don't hesitate to just turn on your mic and ask the question even during the talk. So I'd be happy to try to answer questions. So one of the things that people frequently forget, because Agrobacterium is used in biotechnology so much, is that Agrobacterium is a plant pathogen. In the wild, it infects dicotyledonous plants and some gymnosperms, and it causes a tumorous disease that's called crown gall. And it causes crown gall on many different species of plants, hundreds of different species, including fruit trees, various crops, ornamentals, and so forth. As you could see here on a cherry tree, this is a picture of a crown gall tumor at the base or the crown of the plant. And here's a picture of a crown gall tumor on a rose plant. Since these are soil microorganisms and they require a wound site for efficient infection, the most likely point of entry is going to be on the crown of the plant or below the soil line on the roots. And that's where you find most tumors. So there are virulent strains and avirulent strains of Agrobacterium. Actually, most of the strains that you find in the soil are avirulent. They do not cause tumors, but many strains are virulent, and they are virulent because they harbor one of these types of plasmids. It's called the TI, or tumor-inducing plasmid. These are very large plasmids. They're usually over 200 kilobases with coding capacity for several hundred genes. And there are many, many different types of TI plasmids and related what are called RI, or rhizogenic plasmids, from the related bacterium called Agrobacterium rhizogenes. Now, these plasmids have a number of different regions that they could be broken into. You're probably most familiar with the region called the Tdna, or the transferred DNA region. And this is the region between these left borders and right border sequences, which is destined to go into the plant upon transformation. And in natural TI plasmids, they have oncogenes, which encode for the biosynthesis of auxins and cytokinins, and this is how you get your tumors. And they also encode for the synthesis of unusual compounds called opines. There are many, many different types of opines, and different TI plasmids will instigate the synthesis of different opines. These opines are made from common plant metabolites, such as basic amino acids, alpha keto acids, various sugars, but they're put together in unusual ways because the enzymes encoded on the Tdna will put these compounds together in unusual ways to make these new compounds. Now, plants normally never will make opines. However, and plants can't use opines. However, the strain of Agrobacterium that incites a tumor that makes these opines can catabolize these opines on the TI plasmid, or these genes here, which could catabolize the opines. And most bacteria and most soil organisms cannot utilize these opines. So Agrobacterium is actually genetically engineering plants to make opines, which serve as a food for the bacterium, and very few other organisms in the soil can utilize opines, so it gets an ecological advantage. In order for these virulence genes to be, in order for the Tdna to be able to transfer to the plant, it requires a number of these genes called virulence genes. And I'll be talking about some of these virulence genes in a few minutes. in addition to transferring the Tdna region to a plant, just this region here, the entire plasmid can be transferred from one Agrobacterium strain into another one that may not have a plasmid. So this is called conjugative transfer of the entire plasmid. And why is this important? It's important because if you do not have a TI plasmid, you don't have the opine catabolism genes, and therefore you can't eat opines. But by moving a copy of this plasmid into another Agrobacterium that happens to be sitting in the soil, now the second bacteria, the recipient, has the TI plasmid, and it also can catabolize opines. So it could spread both the tumorigenic principle and the opine synthesis genes, but it will also spread to other Agrobacterium, the genes that can catabolize these opines so that bacteria can partake in the benefits of this reaction. Now, in the 1970s, and then mostly in the 1980s, people started talking about, well, maybe Agrobacterium is actually transferring DNA to plants. And in the late 1970s, this was actually shown to be true. And people showed that the natural Tdna, which contains the oncogenes and the opine synthesis genes, could be found in the DNA, integrated into the DNA of crown gall tumors. But people also learned that there was no gene within the Tdna that is required for transfer of the Tdna. These genes are required for tumorigenesis and for opine synthesis, but not for transfer. The only things necessary for transfer were these border sequences, these left and right border sequences, and genes on the TI plasmid, the virulence genes, and a few genes in the chromosome called chromosomal virulence genes. So because no genes in the natural Tdna are required for transfer, what people could do was they could delete all these genes and then put in any genes they wanted. A selection marker, for example, to know that the gene, that Tdna had been brought into the plant, and any gene of interest you wanted. So this is typically how transgenic plants are made, and for using this substitution of the normal Tdna with artificial genes that we've cloned between the borders. The actual interaction between Agrobacterium and the plant cell is quite complex, and it consists of a lot of different steps. First of all, to be able to have efficient transformation, a wound is important. You can transform plants without wounding, but it's extremely inefficient. So wounded plant cells are, wounded plants try to defend themselves. And unlike animals, they can't run away from their problems. They have to deal with their problems. And so many plants that are wounded make very nasty antimicrobial compounds. They don't sure whether an insect is trying to chew on them, a virus or a fungus is trying to penetrate into it, a bacterium is trying to infect. So they make many of these nasty phenolic compounds. Some of them are called phytoalexins. So these compounds, as well as sugars, the neutral sugars from the cut vascular system, and pectic acidic polysaccharides from degradation of the cell wall, these will be sensed by Agrobacterium, by certain genes, and they will turn on a genetic pathway to start the virulence process. So this process starts by the phosphorylation of this Vir-A protein and the transfer of this phosphate group to another protein called Vir-G. Phosphorylated Vir-G protein then binds to the promoters in the virulence genes and turns on transcription of these virulence genes. So this is what's known as Vir gene induction. Among the genes that are turned on are the genes encoding the proteins Vir-D1 and Vir-D2. And Vir-D1 and Vir-D2 are a site-specific nuclease that see these Tdna border sequences and put a nick in the DNA at these border sequences. And then Vir-D2 is going to covalently attach to one end, the 5' end of this DNA, and peel this off as a single-stranded molecule with Vir-D2 attached. Then what happens is the bacteria have to bind or attach to the plant cell, and Vir-D2 protein, along with several other proteins, Vir-E2, VIR-D5, VIR-E3, and VIR-F, these are called effector proteins, are going to go from Agrobacterium into the plant cell using what's called a type 4 secretion system, which is actually composed of the VIR-B proteins and VIR-D4. Although people talk about Tdna transfer, well, this is actually protein transfer. It's the protein that's being acted on that's going from Agrobacterium into the plant cell through this type 4 protein secretion system. However, because the Tdna is covalently linked to VIR-D2, VIR-D2 will drag the Tdna into the plant cell. Now, once in the plant cell, the Tdna most likely forms a complex with other proteins. One of these proteins is VIR-E2, a single-stranded DNA binding protein, and it's likely that VIR-D2 will bind to the single-stranded DNA, forming this T complex. And although everybody, including myself, believes in this model, this is still just a model. Nobody has actually seen T complexes in plants. Somehow, the Tdna has to find its way from entering into the plant cell and getting into the nucleus. And we think that the Tdna follows the actin cytoskeleton using various myosin proteins to be able to get into the nucleus. And then it gets in using proteins that are called important proteins, important alpha proteins, that bind to these proteins and drag proteins to and into the nucleus. Once the Tdna or the T complex is in the nucleus, these VIR proteins have to be removed. The Tdna doesn't necessarily have to, but it can integrate into the chromosome and where it becomes stably transformed, and then the transgenes encoded on the Tdna are expressed. So this is all a very complex process requiring many different bacterial genes and proteins, but more importantly, probably hundreds of different plant genes and proteins. And one of the things that our laboratory has done in the last 30 years is try to understand what are the plant genes and what are the plant proteins that are involved in this whole transformation process, getting the Tdna, getting Agrobacterium to attach to the plant cell, getting the Tdna translocated into the plant, finding its way to the nucleus, and then eventually integrating into the plant DNA. So I'm going to spend a lot of this seminar talking about the process of Tdna integration, which is one of the things that our laboratory is very actively researching for the last 5 to 10 years or so. And what do we know about Tdna integration? Several things. First of all, we know that Tdna integration into the plant genome is completely random. Now, if you look in the older literature, you may find a lot of papers published on Arabidopsis and in rice and Medicago also, saying that Tdna goes into preferentially into transcriptionally active genes, or to promoter regions, or to AT-rich regions, which tend to be promoter regions. But all of these experiments were done with what we call a selection bias. What people would do is they would find transformed plants, they would see where the Tdna integrated, and in most of the cases, it was in transcriptionally active regions. But in order to get a transgenic plant, you had to be able to express the selection marker on the Tdna. And so if Tdna had integrated into a non-transcribed region of the plant genome, the selection marker would not have been expressed, and you'd throw that plant away. What we did in the around 27, 28 was do this experiment, but without selection for the expression of any genes. So we didn't look at any selection markers. We just looked at plant cells and tried to clone Tdna out of these plant DNA that had integrated. Now, this is, of course, a much more difficult experiment because most plant cells don't have Tdna integrated. And when you do integrate Tdna, every cell is going to have it in a different place. So technically, it was a very difficult experiment at that time. But we were able to find about 115 different Tdna integration events in Arabidopsis. And we showed that the integration is truly random, that Tdna was integrating into genetically inactive regions of the chromosome, such as telomeres, centromeres, heterochromatic regions. And the frequency with which tDNA integrated into these genetically inactive regions actually was the frequency these regions are found in the genome. For example, in Arabidopsis, about 10% of the DNA is heterochromatin, and about 10% of our integration events were in heterochromatic regions. In the late teens, around I think it was 2017 or 2018, Avi Levy in Israel did this experiment again with more advanced genomic technologies that weren't available to us at the time, and came exactly to the same conclusion. Okay, so tDNA integration is random. tDNA often integrates at multiple different places, and at any given place, you could have multiple tDNA molecules at a locus. The other thing that we know, and this is one of the things that our laboratory found a long time ago, is that integrated tDNA could be linked to backbone sequences from vectors. And it's been shown by Bekir Olker in Germany that even bacterial chromosomal DNA could be linked to tDNA in the plant DNA. And recently, we have shown that tDNA from other plasmids in the bacterium could also get into the plant. Now, this is quite a surprising result because people thought that only the tDNA region of the TI plasmid got into the plant. But we now know that other types of DNA can get into the plant also. And this, of course, could cause regulatory problems, because when regulators want to look at plants for field release, they want you to characterize what's in the plant. They want to know exactly what's in the plant. They don't want extra DNA in there. And we're finding that actually much of the time, sometimes up to 5 to 10% of the time, you could find other types of DNA in your plants. So it's very important to do whole genome sequencing of your organism once you want to do field release. So the question is, how does tDNA integrate into the plant genome? What mechanism does it use? So people initially thought, well, maybe it's using some form of homologous recombination. But we know that that's not true, because you could put tens of kilobases of plant DNA into the tDNA, infect the plant, and the tDNA does not use this plant DNA, this homologous plant DNA to integrate. It just goes wherever it wants to, basically. It ectopically integrates. So homology in plants is extraordinarily rarely used. If tDNA is used to infect yeast, however, it will use homologous recombination, but not in plants. So if homologous recombination is not being used to integrate tDNA into the plants, what is being used? Well, the thought is it uses some sort of a non-homologous end joining process. tDNA perhaps integrates either into double-strand break sites or into nicks, single-strand nicks in the genome, using one of two known non-homologous end joining pathways, the classical pathway, the Ku-dependent pathway, or an alternative pathway using perhaps DNA polymerase theta. And I'm going to show you later in the seminar that these pathways may be used, but there probably are some other pathways that are being used also. Or perhaps some other mechanism is being used, a single-strand invasion model. So this figure shows a number of different possibilities of what's happening. And we don't know what's actually happening, but these are some possibilities. So we know that tDNA enters the plant cell as a single-stranded molecule. Maybe it directly ligates into nicks or breaks in the DNA. So maybe this single-stranded molecule directly ligates into the plant DNA. That's one possibility. Another possibility is this single-stranded DNA first replicates into a double-stranded linear form. We know that this happens. We know for sure that tDNA could form double-stranded molecules once in the plant cell. But we don't know whether these double-stranded molecules are the actual substrate for integration or not. Another possibility is that tDNA strands themselves, the actual molecule that's transferred from the bacterium into the plant, this molecule is not the molecule that integrates, but rather it forms a template to replicate a copy of this molecule into the DNA. So it's a replication it's a replicated form of this DNA that gets integrated. And I'm going to talk about this in a moment when I show a model. Another possibility is that once inside a plant cell, Tdna, this double-stranded molecule, can form circles. We know for sure that this can happen. Our laboratory has been able to isolate these Tdna circles or T circles from plants. But once again, we don't know whether these T circles are the actual substrate for integration or whether there's some sort of a dead-end molecule. So we actually don't know which, if any of these forms are the molecule that integrates. So about 5 years ago, a very important paper came out of a laboratory from the Leiden University Laboratories. And they were saying that DNA polymerase theta, a very unusual DNA polymerase, is absolutely essential for tDNA integration into the genome of Arabidopsis. Now, having said that, I'm going to show you data in a little while that says that this is not completely true, that we actually can get tDNA integrated in DNA polymerase theta mutants. But this is the model they presented, and the model is actually very, very interesting. So first thing that happens is there's a double-stranded break in the plant DNA. And frequently when you have double-stranded breaks, exonuclease 1, a 5' to 3' endonuclease, chews back the DNA. So you have these exposed ends. If there's small amounts of homology between the tDNA or complementarity between the tDNA and this end, it may bind here. And this would only be microhomology, needing only maybe 2, 3, 4 bases. Then what happens is DNA polymerase theta could come in and using this plant DNA as a primer, copy the tDNA into the plant DNA. And if it was copied from tDNAs on both sides, you will end up with this right border direct repeat of two tDNAs. And this is actually very frequently found, this type of structure. So this model is very, it's very nice. It explains a lot of things, but it doesn't explain some other things. So do you need breaks or nicks in the plant DNA to be able to integrate tDNA? It is thought that probably you do, but nobody has actually shown this for sure. For example, we know that if you make double-stranded breaks in specific places in the plant genome and then infect the plant with Agrobacterium, tDNA preferentially will integrate at those break sites. So some early experiments done in several laboratories showed that if you put a rare cutting restriction site into the plant genome, for example, ICEU1 or ISCE1, these are very rare cutting enzymes, and then you express the enzyme at the same time you are infected with Agrobacterium, about 1 to 3% of the time, tDNA will integrate at these cut sites. Now, people may think, oh, 1 to 3% is not very high, but it's actually an extremely high percentage of times because tDNA can integrate anywhere in the entire genome. And to integrate at these specific sites to 1 to 3% of the time is actually very high. The other thing we know is during CRISPR editing, where you make double-stranded breaks in the DNA to do CRISPR editing, frequently tDNA integrates at this site. So it's trapped at this break site. So does tDNA integration use one of the non-homologous enjoining pathways to be able to integrate? So before I go on into exploring this, I'd like to review for you some of the non-homologous enjoining pathways that are found in higher organisms and in yeast also. So this picture is taken from the classical non-homologous enjoining pathway in animal cells, but it's very, very similar in plant cells. So in this case, you have a double-stranded break by some sort of damage. This is very dangerous for an organism to have a double-stranded break because you could lose part of your chromosome and then the cell will die. So the organism wants to be able to heal or mend these breaks, to repair these breaks. So when you get these breaks, it sets off a kinase pathway. You use these kinases called ATM and ATR, and they will phosphorylate an unusual histone called histone H2AX. So the phosphorylated form of histone H2AX is called gamma H2AX. And this phosphorylated histone will then bind to both sides of the DNA for maybe hundreds of kilobases on each side of the break. And this will mark a break site and attract the DNA repair machinery. In the classical pathway, a heterodimer of Cu70 and Cu80 will then be attracted to this site, and they will bind to the DNA, protecting the ends from further resection by endonuclease. And then this will attract other proteins then that are involved in repair, DNA polymerases, and then ligase complexes that will ligate the DNA back up. So you end up with double-stranded DNA again, but there may be mutations that occur at this point. It is more important for the organism to be able to fix this DNA, even with mutations, than to not have mutations. Of course, it wants to fix the DNA without mutations, but that would be by homologous recombination. And that's only available in certain parts of the cell cycle in certain cells. So other cells have to use this non-homologous end-joining pathway. It's better to have a mutation here, than to have DNA that's not repaired, because the cell's going to die if the DNA is not repaired at all. Now, there's a second pathway or second set of pathways called the alternative pathway that use a different set of enzymes to be able to repair DNA. They use the PARP1 and 2 proteins, the MRN complex, the Werner helicase, and DNA ligase 3, which is actually not found in plants, but this is a slide taken from animal systems. And they will use this other pathway to repair the DNA. But this pathway is very, very highly mutagenic because it uses small amounts of microhomology. And if you resect the DNA ends that are still available will search for homology throughout the genome. And it may find homology in another part of the chromosome, or it may even find homology on another chromosome. So this will eventually end in chromosome rearrangements, inversions, translocations of chromosomes, and it could be quite dangerous. And this is actually what happens in a lot of cancer situations. And this is mediated by DNA polymerase theta, and DNA polymerase theta is currently a cancer target, a therapeutic target for cancer. Interestingly enough, a lot of the types of misrepair that you see using this alternative non-homologous end-joining pathway, these inversions, these chromosomal translocations, a lot of deletions, are exactly what you see when tDNA integrates into the genome. One thing that most people don't understand is that when tDNA integrates, it frequently causes a lot of chromosomal rearrangements. In one study, it was shown that 19% of the tDNA integration events that were occurring in Arabidopsis, 19% were chromosomal translocations. And why didn't people know this before? Because they didn't look for it. So you sequence one side of the tDNA into the plant DNA, you sequence the other side of the tDNA to the plant DNA, but to see this translocation, you have to sequence the plant DNA all the way through the tDNA into the other side of the plant DNA. And this can now be done by these long-range sequencing types of techniques. And what you sometimes find is DNA from chromosome 1 linked to tDNA, and on the other side, chromosome 3. Okay, so these types of translocations and all sorts of chromosome rearrangements have now been shown to be actually very common when tDNA integrates. Another reason when you try to put transgenic plants out into field for field release, that you should do total genome sequencing rather than just depend upon looking at the junctions at a very short distance. So I'm going to talk now about some work we did in our laboratory on trying to understand the process of tDNA integration. But before we get into the actual data, does anybody have any questions they may want to ask? So if you do, please just unmute yourself and go ahead and ask some questions. No questions? Okay.
Question on the chat, yes.
Go ahead, please.
Yeah.
Go ahead.
I'm sorry, I have a throat problem, so I write my question on the chat if I may.
Yes.
Yes, but do I have access to the chat when I'm in this mode? I don't think I have access to the chat unless I go out of this presentation mode. So why don't you just read your question?
Yeah, okay. I can help to read the questions. Prof. Gelvin, thank you for your talk. I personally did Agrobacterium mediated transformation to Arabidopsis, but I feel lacking in basic understanding about it. I am very much interested to the theory of Tdna integration to plant genome. What I understand from your lecture here are the basic requirements of the integration. First, double strands break on the plant's DNA strands. Second, Tdna strands which will attach to a few exposed bases on the Tdna strands, the DNA strands. And the third, endonuclease delta or other pathway of DNA repair. Kindly confirm if my understanding is correct. My question is, what induced the double strand break in plant genome, as it is the first requirement of the integration? Regards. The question is from Dr. Sinto.
Okay, so very good question. What you said was basically correct as one possible model, although we still don't know for sure whether this is true. And I'm going to talk about, I'm going to talk a little bit more about this in a few minutes. In terms of what causes double-stranded breaks, that's a really interesting question. Double-stranded breaks happen all the time, randomly. An organism has to repair them very quickly because it's really important to do this, or else you're going to end up with losing parts of the chromosome. One possibility, and this has been shown with other phytopathogens, but it hasn't been shown with Agrobacterium, is that when the phytopathogen attacks a plant cell, it induces double-stranded breaks as a damage response by the plant cell. This has been shown by other pathogens, both bacterial and fungal pathogens, that you can get double-stranded breaks resulting from essentially a PAMP response, pathogen-associated molecular pattern response. We did some experiments a number of years ago where we asked the question, does just exposing a plant cell to Agrobacterium cause double-stranded breaks? And our results, unfortunately, were inconclusive. We thought we were seeing more double-stranded breaks with Agrobacterium than without Agrobacterium, but it was not significant, statistically significant. So the question is still an open one, but I would still wager that Agrobacterium induces double-stranded breaks. If not, it probably takes advantage of some double-stranded breaks that are occurring all the time during replication and things like that. That's my guess. But it's a really good question in an area that really needs a lot of work. Any other question, or should I go on?
There is the Pak Akwin Polosoro, Pak Darda.
Okay.
Raise hand.
Okay, Pak Akwin, please.
Okay, thank you very much, Prof. Darda. And it's very nice presentation for Prof. Gelvin. It's very interesting, but I have two questions. The first question is, there is any study that concern to the flanking sequence? So I believe there is a lot of event in transgenic plant, right? So right now, big data is very a study about big data is really very improvement in here. So if we can, there is any study, they collected the flanking sequence and recognize, probably there is a recognized sequence or any, what you said, the endonuclease function related to the insertion in the Tdna.
Are you asking, is there any particular sequence,
Yes.
short sequence motif that a Tdna goes into? As far as I know, no. There's no known particular sequence motif that Tdna will use.
There's any study for that before?
Well, there have been tens of thousands of Tdna, plant DNA junctions sequenced, and there's just nothing that comes out as being a consensus for any type of motif. You know, there have been tens of thousands of these done in Arabidopsis and in rice and Medicago. And nothing really has come out as anything of a particular sequence motif. One thing that may be interesting, but I don't know the answer to this, is there a particular type of chromatin structure that Tdna integrates into? But I don't know the answer to that, okay? I don't think that experiment has really been done.
Okay. And,
okay.
I have one more question. So I've been working for plant transformation for years. So there is, I don't know, it's very complicated because there is a lot of progress from the plant. So the efficiency of transformation is related to the backbone factors. So it's related to the plasmid and also related to the Agrobacterium strain. Do you have any suggestion how can we choose the right plasmid and right Agrobacteriums for the better efficiency for plant transformation?
That's a really good question, but you know, some Agrobacterium strains are more highly virulent than others, but it's on certain plants. So for example, what we call EHA 101, EHA 105, that you may be familiar with those strains. It's an agropine type of TI plasmid. Sorry for all the rest of you, we're getting into details here, but that strain is extremely virulent, highly virulent on solanaceous plants, but not as virulent on like Arabidopsis, for example. However, in Arabidopsis, another frequently used strain, a different type of strain with nopalene type genes, GV3101, you may be familiar with that strain, is very, very virulent on Arabidopsis roots, but not as much on solanaceous plants. So it appears that we don't know why particular plants are susceptible or virulent, and basically you just have to try different combinations. We do know that in some cases, there's a gene on some of the nopalene plasmids called TZS, which is involved in cytokinin biosynthesis, and that gene may play a role in virulence, but it's certainly not the only thing. So I wish I can give you the magic bullet of which exact plasmid and which exact strain is good for your plant, but until we start really understanding much better what is it that makes some plants resistant and susceptible, until we have a much better molecular understanding of that, I think it's just going to be more of a trial and error sort of thing.
Okay. Thank you.
Okay, I'm going to go on with my lecture now. Okay. So I'm going to present some work that we've done in our laboratory looking at Tdna integration. So let's look at some of these non-homologous end joining proteins. What's their role in Tdna integration? So some labs showed that if you mutate non-homologous end joining genes in the plant, you get a slightly decreased stable transformation. And some labs showed if you mutate these exact same genes, you have virtually no effect on transformation. But none of these studies actually looked at Tdna integration biochemically, looking at the molecular aspect of Tdna integration. They only looked at reporter assays, okay? Transformation is usually reported as resistance to a selection antibiotic, for example. So if Tdna had integrated into a dead region of the genome and therefore could not express the selection marker, you would think that it had not integrated when it actually had. So our studies have shown that mutation of these non-homologous end joining genes does not decrease either stable transformation or Tdna integration. In some cases, it actually increases these processes. So this is quite a surprise. I'm going to tell you the data for this. So first of all, I'm going to show about how we do these assays. Our laboratory is a basic science model systems lab. We use mostly Arabidopsis, sometimes tobacco, and occasionally we've done some work with rice. So this is our favorite assay. These are roots of Arabidopsis. We cut the roots into lots of, hundreds of little pieces that are 3 to 5 millimeters in size. We inoculate them with Agrobacterium, and after a few days, we spread each of these root segments out on a plate with antibiotics to kill the bacteria, and whatever sort of selection we're going to use to look for tumors, for transformation. In this case, the selection is for tumorigenesis. There's no hormones in the medium. Normal roots will not grow on this medium, but tumors will. And sometimes we work with plant mutants, and sometimes we find mutants that cannot get very good stable transformation. And we call these rat mutants for resistant to Agrobacterium transformation. And we have about 150 of these mutants now in different genes in Arabidopsis. Now, we could also do the opposite. We could deliberately infect wild-type plants with a low concentration of Agrobacterium and deliberately get a very, very bad infection and look for mutants that are hypersusceptible, called hat mutants, or hypersusceptible to Agrobacterium transformation. And we have a number of these mutants also. So when you mutate the gene, you get a higher level of transformation. So what we did was we took these various Arabidopsis genes involved in non-homologous conjoining, the classical pathway or the alternative pathway, and we took mutants of these genes. We showed that these genes were not functioning, and then we did stable transformation. As you can see here, 1 is the wild type level. We never saw a decrease in transformation with these different mutants. And with several genes, if we knock out Q80 or Q70 or a plant-specific DNA ligase 6, we actually got a 2 to 3-fold increase in transformation. So this was really surprising, because if these genes are important for transformation, if you knock them out, you should get less transformation. We never saw less transformation. Now, there's another type of transformation that people look at also, and this is called transient transformation. So in transient transformation, Tdna gets into the nucleus and it becomes replicated to a double-stranded form, but it is not integrating. So it's expressing its transgene from a non-integrated form, and this could happen for a few days until the DNA just eventually goes away. Okay, it gets nuclease digested or it gets diluted away and has not integrated. So in this case, we're looking at expression of a Gus transgene, and this, we stain the root segments after 6 days with X-gluc, and if the Tdna had gotten into the cell, it makes a blue color. Here's a wild type. And here's a rat mutant. Remember, it can't integrate Tdna very well, but it could still express very well, at least transiently. So what this suggests is that the Tdna had gotten into the nucleus and it had become double-stranded, but it couldn't integrate, or it had integrated and silenced. In this particular type of rat mutant, we see that transient transformation is also very much decreased, suggesting that the mutation occurred very early on, or the block point for transformation occurred very on. Perhaps the bacteria didn't bind well to the plant cells, or it didn't transfer Tdna, or the Tdna had not gotten into the nucleus. And we have a number of different mutants with this. Now, if you look at transient transformation should not be affected by any of these mutations in these genes involved in integration, because transient transformation does not require integration. And that's exactly what we saw. We saw no effect on transient transformation with these various different mutants. Now, most laboratories at this point stop and say, oh, we're looking at integration. But you're not looking at integration in these assays. You're only looking at stable transformation, which could or could not reflect stable integration. So we went the next step and actually looked molecularly at integration. What we did is we infected Arabidopsis roots, grew calli from these roots without any selection at all, isolated DNA from these calli, put the DNA on a gel, and probed with a Tdna gene. In this case, that was in the Tdna, this is a Gus gene. You can see this is high molecular weight plant DNA, so this is a DNA blot, but there's no restriction nucleus digestion. And now we're looking without any selection for the presence of Tdna that's integrated into high molecular weight plant DNA. And we see it. In fact, in the Q70 and the Q80 and the DNA ligase 6 mutants, we see 2 to 3-fold higher levels of Tdna integrated into the plant DNA, which is exactly the same thing we saw with a 2 to 3-fold increase in stable transformation. So this is a molecular characterization of what we were seeing with the stable transformation results. So then we started looking at higher order mutants. So this was stimulated by a very nice paper that was published by Charles White's group. And what they showed was that if you have single mutants, these are all mutations in various DNA repair pathways, in the classical pathway, non-homologous gene joining pathway, the alternative pathway, or homologous recombination. What they did is they looked at a number of single mutants, double, triple, and quadruple mutants, and they looked at transformation. And what they saw was, what they said they saw was, as you had more and more mutations in these different DNA repair pathways, you got less and less transformation. But you always got some transformation. It wasn't zero. Now, this was very peculiar because they said you got a 2.5-fold decrease in transformation with the Q80 mutant, and we said we had a 2 to 3-fold increase. So this was very curious. We asked Charles White if he would please give us the mutants that he worked with, and he very nicely gave us these mutants, and we started working with the same mutants that he did. The first thing we noticed was that his DNA repair mutants were sick. So if you try to germinate the seeds, the more mutations you put into these, knocking out these different pathways, the poorer the germination was. Also, the roots were getting smaller and smaller, the plants were getting smaller. This is not unexpected, because if you destroy genes that are involved in DNA repair, you can't repair any mutations that occur. So over the generations, you're going to start piling up more and more mutations that you can't repair. And the plants get sicker and sicker because of this. So the fact that the Tdna, that he was seeing less transformation could occur not because the mutations of these genes were important for the transformation, it was just purely because the plants were sick. And this is what we think was happening. Because if you look at transient transformation, you see that transient transformation is decreased also in these mutants. Now, Charles White's group didn't see this initially because he used very high concentrations of bacteria to inoculate. He used more than 10 to the 8th CFU per mil. But he was saturating the reaction. And as you dilute the bacteria, you start seeing, yes, these plants are not transiently transforming very well. So what happens if you stably transform these plants? Turns out you don't see that much drop. You see less than a 2-fold drop in stable transformation, even when you knock out 4 different DNA repair pathways. We did another assay also to look at Tdna integration. This is a technique called droplet digital PCR, which is an extremely sensitive technique. You could see about 1 Tdna molecule per 100 Arabidopsis genomes. So again, we infected the roots of Arabidopsis, we grew cali without selection, we isolated DNA, and then looked for Tdna that's integrated into this plant DNA. And you could see that between about 1 in 20 to 1 in 40 plant genomes had a Tdna integrated in the wild type, but also in the mutants also. So despite the fact that you were knocking out all these different DNA repair pathways, you're still getting very good Tdna integration as assessed by this actual quantitative method. So as I said before, a very important paper came out from the Horticulture and Teisterman group in 2016, where they said that DNA polymerase theta mutants in Arabidopsis, these are called TEB mutants for Tabichi, okay, cannot be stably transformed. They said DNA polymerase theta was absolutely required for transformation. So at about this time, my wife and I went to Seiichi Toki's laboratory in Japan, and they've been working on a lot of DNA repair mutants in rice. So we worked with Ayako Nishizawa Yokoi and Hiroaki Saika, and in Seiichi Toki's lab, and Lenning is my wife. We went to, she's the lead scientist in our laboratory. And so we went to his lab, and we found all these DNA repair mutants in rice that they had made, and we asked them, had you ever tested these mutants for transformation? And they said no. So that's what we did. We tested these mutants for transformations, both transient and stable transformation. Now, they had a very nice reporter system. The Tdna had a constitutive promoter that was driving the expression of a GFP gene. Okay, this is a GFP blastocytin resistance gene. This gene would work either for transient or stable transformation, okay, and a luciferase gene, which had a minimal promoter and will not function unless Tdna integrated into a region of the plant genome close to an enhancer or an activator, and then it would activate this expression of this luciferase gene. And that's what you see here is this luciferase gene, luciferase activity in the wild type plants. So this is what's called an enhancer trap vector. We were really surprised when we started assaying his DNA repair mutants, his rice DNA repair mutants, and we saw that in the DNA polymerase theta mutant, DNA polymerase theta is also called polyQ, we saw Tdna integration. Okay, not as good as wild type, but we saw pretty good Tdna integration. Now it turns out that these rice mutants in DNA polymerase theta had a phenotype, and that is they would not regenerate. So normally you regenerate rice from cali on regeneration medium. This is wild type rice. And here's the polyQ mutant. And if you put it on regeneration medium, it will not regenerate. This has nothing to do with transformation at all. It's just straight regeneration. So polyQ mutants have a developmental phenotype. However, we found that if you transformed these cali with a construct that contained a polyQ gene, the transient expression of this polyQ gene was enough to be able to get regeneration of this plant. So if we didn't have a polyQ gene in there, if we had a Gus gene in there, you couldn't get regeneration. But if you transiently expressed a polyQ gene, all of a sudden you could regenerate plants. So this left us with a protocol now for getting transgenic rice plants in polyQ mutants rather than just rice cali. So the idea was to generate transgenic polyQ mutant rice cali. We could isolate and characterize the Tdna plant DNA junctions from these cali. Then we could retransform these cali with a second Tdna containing a wild type rice polyQ gene, regenerate rice plants from this, and then characterize the regenerated plants to see this first Tdna if it were still in there. And this would then prove that in a polyQ mutant, you could actually get Tdna integrated and make a plant out of it rather than just cali. And so this is work that Ayako did. It's not published work yet, but we're finishing this work up right now. She found, for example, Tdna integrated into callus DNA. Here's what the Tdna looks like. There's plant DNA, then plant Tdna, then plant DNA. She characterized these borders. She found microhomology. This is in a polyQ mutant. Then we were able to regenerate this plant and show that this original Tdna was in the regenerated plant. So what did the Tdna junctions, the Tdna plant DNA junctions look like in a DNA polymerase theta mutant? They look exactly like the junctions you see in a wild type plant. You see deletions at the Tdna left border. They're more frequent and more extensive than Tdna deletions at the right border. This is what you see in wild type infection, wild type plants. There's frequently filler DNA that's found between the integrated Tdna and the plant DNA. The filler DNA could come from within the Tdna. It comes from the Tdna backbone. It could be plant DNA from some other place that's been put in between this. We see this also in the polyQ mutant. And most importantly, we see the use of microhomology between the Tdna border region and the plant pre-integration sites. Now, microhomology is supposed to be used by DNA polymerase theta, but these were DNA polymerase theta mutants, and they were still using microhomology to integrate Tdna, which means that there must be yet some other way of using microhomology to integrate Tdna, some way in addition to DNA polymerase theta. So when we got back from Japan to Purdue, my wife said, why don't we take a look at those Arabidopsis mutants that the Leiden group did? So we ordered the same mutants, and we showed that you could get stable transformation of these Arabidopsis roots. It was not as good as wild type, and the cali grew much slower, but that's because these plants, the callus would grow slower even if you didn't transform it, if you just try to grow callus. So also in Arabidopsis, as well as in rice, you see there's a developmental and growth phenotype by having a DNA polymerase theta mutant. But we were able to isolate junctions from these calli. We isolate DNA from these under non-selective conditions. We were able to look at Tdna integration using droplet digital PCR. This was done by Hiroaki Saika in Japan. They helped do these experiments. And we found that in these DNA polymerase theta mutants, they still integrated between 50 and 90% of the amount of Tdna that you see in a wild-type plant. So therefore, DNA polymerase theta is not required for Tdna integration. Although I am not saying that it may not participate in the process. It may participate in the process, but we've now shown both in Arabidopsis and in rice, it's not required. It's not absolutely required. There must be something else that's going on. So more recently in our laboratory, Dr. Chaoling Zhang in our laboratory, a postdoc has done some experiments in which we tried to see which part of DNA polymerase theta was important for transformation. So what she did was she expressed in transgenic Arabidopsis different domains of the DNA polymerase theta protein. DNA polymerase theta is a very, very large protein, over 200 kilodaltons, and it has a very unusual properties. It has a helicase as part of the same polymerase protein. That's unusual. That's not found in other polymerases. It also has other properties that are very unusual. It template switches. It starts copying one part of the genome, then it jumps to something else, which is very unusual for a DNA polymerase. It's also known as a bypass polymerase. It will bypass damage in the DNA, and because of that, it will stick in some random bases, so it's a low fidelity polymerase. So we wanted to know if we overexpress the full-length polyQ, DNA polymerase theta, would it enhance stable transformation? Or if you overexpress some of the peptide domains, would it inhibit transformation? This would be a dominant negative effect. So we first expressed either an empty vector or just the polymerase, DNA polymerase domain of polyQ or the full-length polyQ. It had no effect on transient transformation and had no effect on stable transformation. She looked at many, many different transgenic lines and she saw no effect at all. However, when she expressed the dead box region, which is part of the helicase domain, she inhibited stable transformation by a factor of about 2, but it did not affect transient transformation. So this is stable transformation. This is the control. This is the full-length polyQ. It's actually a little bit extra. It's a little bit higher, but in the dead box domain, we inhibit transformation by about 50%. So what we conclude from this is that DNA polymerase theta is involved in Tdna integration, but it is not absolutely required for Tdna integration. So I'm going to conclude this part of the seminar, and then I'm going to go on to a genome editing part of the seminar. So in conclusion, none of the tested non-homologous end-joining genes is required for either plant somatic cell stable transformation or for integration, which we've shown by molecular methods. DNA polymerase theta may contribute to stable transformation and may contribute to Tdna integration. However, it is not required for use of microhomology, and also it is not absolutely required for integration. So before I start the next part of the talk, which I promise is going to be shorter than this part, do you have any questions that you would like to ask me about this part of the talk? The conclusion is we don't know what DNA repair pathway and enzymes is responsible for integration. Either multiple pathways could be used. If you can't use one, use another, or there's some yet unknown pathway that we need to discover. That's our conclusion from this. Any questions for this part, please?
Yeah, any question? Or a short question for Gelvin. So what's the maximum loads of the plasmid for the Tdna? So how long the Tdna we can put between the left border and right border that can be transferred? Or how many genes we can put in that location?
Many natural Tdnas that are normally found in TI plasmids are 20 to 25 kilobases. So that's 20 to 25 genes or so, 20 genes. You can make Tdnas that are very, very large, up to 50 to 100 kilobases. They can transfer all those genes, although it's less efficient, and frequently the tDNA doesn't transfer all of them. But you could easily transfer 10 to 20 kilobases of DNA with no problem whatsoever.
For the commercial transformation or the commercial GMO, how many genes that at once transfer from Agrobacterium to the plants?
In theory, as many as you could put in. Okay.
In theory,
People have done 5 to 10 genes. Okay.
5 to 10.
Yeah, people have done that. Trying to put a whole pathways in, for example. So for new biosynthetic pathways, people have done that. So of course, as you add more and more genes and the tDNA gets larger and larger, the efficiency goes down. Sometimes you don't get all the genes in. But you can get a lot of genes in.
Okay, one more question. So in the integration of the tDNA from the cytoplasm into the nucleus, who's play the most roles? Is still a Agrobacterium, or it's the plant cells itself that uptakes the tDNA from the cytoplasm to the nucleus? Yeah.
We think that at this point, once you're in the plant cell, it's mostly plant genes and enzymes and proteins that are involved. Although there are a few, there's only 5 different effector proteins Agrobacterium puts into the plant cell. We think that those proteins do play some role, but the major role is played by plant proteins. For example, we have over 100 different plant genes, mutations in 100 different plant genes that are that affect transformation. We know from some work we've done recently, we haven't published yet, that the actin cytoskeleton and multiple different types of myosins are involved in trafficking tDNA to the nucleus. We also have done some mass spec analysis where we've looked at how some of these Agrobacterium proteins that are transferred to the plant cell, what proteins in the plants that they're interacting with. And there are many, many plant proteins that are interacting with. So we think that for the most part, after tDNA gets transferred to the plant cell, it becomes a plant cell biology problem for the most part at that point.
So also the case with transient and stable transformation is controlled mostly by,
yeah, it's the only difference between transient and stable is stable, you have that extra part of tDNA integration. But, you know, getting it to the nucleus, becoming double-stranded, it's all the same. It's just a matter of whether you're going to then integrate. The thought is that the vast majority of tDNA that gets into the plant and that gets into the nucleus doesn't integrate. Only a small part of the tDNA, only a small minority of the tDNA molecules that get in eventually integrate. That's what the thought is. But nobody's been able to actually quantify this. I don't quite know how to do the experiments.
Thank you.
Okay, so I'm going to, is that okay? Should I go on then?
Yeah, sure.
Okay, so I'm going to talk to you now, the second part of this seminar, about some work that we've been trying to do to engineer Agrobacterium to make it more useful for genome engineering. And this is work that's been done by Dr. Lanning Lee in my laboratory. And what we're trying to do is integration-free tDNA transfer by VRD2 mutants. In other words, we're trying to get tDNA efficiently into the nucleus and express, but not have it integrate. Because that way you don't make a transgenic plant and there's no problems with regulations for transgenic plants then. So there are circumstances where it may be desirable to avoid integration. For example, if you're introducing correction templates for homology-dependent repair, you want to switch one region of the DNA for another. You don't want the DNA that you're putting into the plant to go randomly in. You want it to only to switch by homologous recombination. Also, if you're introducing genome editing reagents into the plants, you want to make an edited plant. You want the Cas protein and the guide RNAs to express and to make whatever mutation you're trying to make in the plant, but you don't want the tDNA that has the genome engineering genes to integrate. This is usually done now. That's how this is done. You introduce with Agrobacterium a tDNA with your encoding your Cas protein and your guide RNA. It integrates into the genome. You look for the plants that have your edits, And then to get rid of the tDNA, you segregate the tDNA out. That's what's commonly done now. However, segregation is time consuming. Depending on the plant, it could be very costly. And it can be difficult or almost impossible for plants with long generation times, for example, trees, or species that are vegetatively propagated, banana, potato, sweet potato, for example. So if the tDNA integrates, you're not going to be able to segregate it out very easily. So wouldn't it be nice to be able to get the tDNA in, express the CRISPR transgenes, have it do its work, and then just disappear? So what we're trying to do is develop this new technology we call integration-free tDNA transfer. And we're using several different approaches. One approach that we're using is to identify mutations in VIR-D2. This is an Agrobacterium protein that unlink tDNA transfer from integration. So how does this work? Okay, so this is, pretend that this region, this double-stranded DNA is part of the TI plasmid, and here's the tDNA left border, and here's the tDNA right border. VIR-D2, along with VIR-D1, comes in and it nicks one of these strands on both sides in the border, and then VIR-D2 covalently links to this DNA, which is peeled off. The single-stranded tDNA is peeled off as a VIR-D2 single-stranded molecule. Once it's in the plant cell, we think, although nobody's actually seen this, that the T strand then associates with VIR-E2 protein, which is another protein that's transferred to the plant from Agrobacterium. And this is called the theoretical T complex. And this T complex, the VIR-E2 protein, we think is important for protecting the tDNA from nucleases as it goes to the plant nucleus. That's the model. Now, why do we think that VIR-D2 is important for integration? VIR-D2, of course, is essential for tDNA processing. VIR-D2 is the protein that leads the T strand out of Agrobacterium through the type 4 secretion system. So it's essential for transfer. And VIR-D2 also has very strong nuclear localization signal sequences in it. So VIR-D2 is very important for getting the tDNA to get to the nucleus. But why do we think that maybe it's involved in integration also? Well, a number of years ago, a friend of ours, Walt Ream, made this mutation in VIR-D2, which we call the omega mutant, or he called it the omega mutant, because it's in this very tail end of the VIR-D2 protein. Here's the VIR-D2 protein map of it. And he replaced these conserved sequences, DGRGG, with 4 serines. And what happens when you do that, and we tested this in our laboratory along with Walt's help, was that you see that transient transformation is reduced 4 to 5-fold, but stable transformation is almost completely eliminated. Okay, it's down 50-fold approximately. So you preferentially decrease integration, although you do decrease transient transformation also. So that's one of the reasons, that's the starting reason why we think that the omega region in VIR-D2 is involved in integration. So VIR-D2 is important for processing in the bacteria, for transfer to the plant, and also integration. And the thought is maybe we can uncouple these major functions. Could we identify VIR-D2 mutants that maintain only tDNA processing and transfer functions, but lose the integration function? And this would be very useful for making plants that are edited but don't integrate the tDNA into the DNA, the genome editing reagents. So this is how we went about doing this. This is a part of the TI plasmid, okay? This is the TI plasmid here. Excuse me, this is the TI plasmid. And on the TI plasmid is this VIR-D operon. It's only part of this large 200 kilobases per plasmid. And what we did is we made a non-polar deletion of VIR-D2, in which we deleted most of the internal part of VIR-D2, but left the amino terminus and the carboxy terminus in frame. So it makes essentially a dead protein. And therefore, it's non-polar on VIR-D3, VIR-D4, and VIR-D5. For those of you who remember your microbial genetics, you have to watch out about polarity. So there's a promoter, expresses VIR-D1, VIR-D3, 4, and 5, but it makes an inactive fragment of VIR-D2. Then what we did was we took this region here and put it on another plasmid that has the promoter for VIR-D, VIR-D1, and VIR-D2. And then what we did was we used mutagenic PCR to make random mutations all over the place in this VRD2 gene. And we sequenced more than 1,000 of these random mutants. And this is what we found. This is only a small number of the mutations that we got. The top sequence in each line is a nucleotide sequence encoding VRD2, and the second sequence is the amino acid sequence. And anytime you see something in red means that we have a mutation there, either a mutation in the DNA or in the protein. Sometimes mutations in the DNA do not cause a mutation in the protein. Sometimes you will get an early stop codon. There's one someplace over here, I can't remember. Yeah, there's an early stop codon here where TGG went to TGA, I think, remember. And so, but the important thing here is that if you look at this, the mutations are random throughout the genome. So each molecule, each clone we have of VRD2 has one or at most two different mutations in it. And then what we did is we tested each of these VRD2 proteins to see how it affected transformation. So we had 1,000 of these or so. How did we do this? We took our Agrobacterium strain that had a non-polar VRD2 mutation, so there's no VRD2 here. It also has a Tdna binary vector in here that can have a Gus gene and kanamycin resistance, and it's a virulent strain, so it has oncogenes that will make tumors. And we put into this strain each one of these different mutant VRD2 proteins we have. So we had, I don't know, somewhere around 300 different mutations in VRD2 genes that we tested, and we put this in here. Then we infected kalanchoe leaves. It's an ornamental, and I'll show you in a minute what this looks like. And because there's oncogenes, we asked for tumors. Could it make a tumor? If it can make a tumor, then we're not interested, because that means the Tdna had stably integrated. So we took all the mutants that could not make tumors and tested them for transient transformation in tobacco leaves through a tobacco leaf infiltration, looking for Gus activity. What we were looking for was good Gus expression, meaning good transient expression, but no tumorigenesis, meaning no Tdna integration. So we found ones that integration was essentially zero or very, very low. Then they still had good transient transformation. Then we used a more quantitative Arabidopsis assay to actually quantify what we were doing. And what we're looking for is VRD2 mutant strains that have no stable, but retain good transient transformation efficiency. So this is the assay I was telling you about. There's this, it's sometimes called mother of thousands, Kalanchoe digramontiana. The leaf is, I don't know, 10, 15 centimeters long. And you can make lots of different wounds in the leaf. And in each one of these wounds, you put a different strain of Agrobacterium in, wait for a few weeks and see if you get tumors. Here's a positive control with a wild type VRD2. Here's a negative control, no tumors because you have no VRD2 in here at all. Some of our VRD2 mutants still made tumors. We're not interested in them. But here are two strains of Agrobacterium with mutant VRD2 genes that don't make tumors. So we're interested in those. So we take those and many other ones that didn't make tumors, and then we infiltrated tobacco leaves, and 24 hours later, you could stain the leaf with X-gluc to see whether you have transient Gus activity. Now, in some of the leaves, we had no transient Gus activity. That just means basically we completely killed VRD2. We're not interested in those. But some of them still had pretty good Gus activity. Here's wild type. Here's the negative control, the positive control. Some of them still had pretty good Gus activity. So remember, these came from no tumors, but still pretty good Gus activity. Then we did quantitative analysis and found some of these lines, although they had very, very low tumorigenesis activity in Arabidopsis, extremely low, still had up to maybe about 50% of the level of transient activity as the wild type did. So they weren't as perfect as wild type for transient, but they were still pretty good. So the next thing we wanted to know was, well, how can we use these for genome editing? So now we have our non-polar VRD2 mutation in the TI plasmid. We put in our various VRD2 mutants, and now our Tdna binary vector on the Tdna has a Venus gene, which is a polyfluorescent gene, and a Cas9 gene and a guide RNA. And this we're going to use for editing. And what we're going to do is we're going to infiltrate tobacco leaves. wait for 4 to 6 days. We know that the infection worked well because the Venus gene is in there, so we could see this Venus fluorescence under a fluorescence microscope. Then in this region that is fluorescing, we isolate DNA from this region. We amplify the DNA surrounding where our CRISPR cut site should be. And then we conduct wide-seq analysis to look at the percentage of time specific nucleotides have been mutated by CRISPR. And then we analyze the types of mutations. So this is an example of what we're looking at. So this would be our PAM sequence would be over here. This is our cleavage site, +1 and -1. So CRISPR Cas9 would cut here. And now what we did was we looked at all the different molecules that we'd amplified from this region. There were thousands of molecules. We did wide-seq analysis, and you could say, what percentage of time do you see a mutation at each of these different sites? And in this particular one, which is very good, 5 to 10% of the time, we saw mutations in this cleavage site around here. Remember, there is absolutely no selection for the presence of Tdna. So this is very, very good with no selection. The control was we used a strain similar to this, but it had no guide RNA, and we got essentially no mutations. So that's our negative control. So then what we did was we looked at various different VIRD2 mutants. Here's our PAM site. You cut 1, 2, 3 bases upstream from the PAM. So the cut site is right here. And now we're saying, what percentage of the time, when you're looking at thousands or tens of thousands of amplicons, what percentage of the time do we see mutations with wild-type VIRD2? In this case, it's 1 to 2%, and various mutants. And we see not as good as wild-type, but some of them are not too bad. This one here actually is pretty good. This mutation was we called 4E12, okay? It's about maybe 1/4 to 1/3 of the efficiency of wild-type VIRD2. So in the process of doing these analysis, we found a mutation which we call 4FO3 that's actually hypervirulent. It makes more tumors than normal. So we thought, oh, this is interesting. Maybe we could take the mutation from this one, combine it with the mutations we had before that didn't integrate, and we could increase our transient transformation but still keeping our stable transformation 0. And this is exactly what we have. Here's our wild type, looking at tumors. 4FO3 gives a slightly elevated transformation for tumors. The 4E12 mutant that I showed you before doesn't have very good integration, no stable transformation. And 4FO3 also does not have good integration. So then, will this combination of mutations now enhance CRISPR editing efficiency? And the answer is yes. So if we look at editing of these bases around the cut site with wild-type VIRD2, then if we look at our 4E12, which was about 1/3, if we look at our 4FO3 mutant, which is hypervirulent, we get higher levels of CRISPR of mutation. And if we combine it, so if we put 4FO3 plus 4E12 into the same VIRD2 molecule, we then raise our mutagenesis from 4E12 to about half of what we see in wild type. So now we've gone from about 1/4 or 1/3 of the wild type level to about 1/2 the level. We also tested this to make sure our double mutant still has very, very low stable transformation, and it also has about half the level of wild type transformation. And in the process of doing this, we also found a very unusual rearrangement of VIRD2, which we call the TR mutant. And this TR mutant also has very, very poor stable transformation, so it's not integrating Tdna. But it's also doing a very good job in CRISPR editing, about half the level of you see in the wild type. So some VIRD2 mutants get edited well without mediating Tdna integration. So we don't have the perfect VIRD2 mutant yet. The perfect one would be wild-type levels of transient transformation and wild-type levels of editing, but essentially no stable transformation. We have mutants that have almost no stable transformation and about half the level of transient transformation and half the levels of editing of wild type. So whether that's going to be good enough, there's a trade-off here. It doesn't edit quite as well as wild type, but it has essentially zero stable transformation, so you're not getting Tdna integration. So to conclude this section, we use mutagenic PCR to alter the sequence of VIR-D2 and Agrobacterium protein. We obtain mutants that could not stably transform plants. Some of these mutants continue to support reduced efficiency, about half, suggesting that sub-Tdna had entered the plant but had not integrated. We started testing these mutants for their ability to mediate delivery of CRISPR reagents and to support editing. And we now have some evidence with some of these mutants that we can get editing at about half the frequency of a wild type 32 gene, but with the absence of Tdna integration. Okay, so that's the end of my talk right now. And I'm going to stop sharing my screen. Is that okay? Or do you want to, well, I'll keep sharing my screen in case you want me to go back on slides. Okay. So if you have any questions now, I'd be happy to talk, to answer any more questions you have about the work that we've done.
Thank you, Prof. Gelvin. In the chat, there's some questions and also just suggestion. First, from Dr. Sinto. This is not a question, I think.
It was just not my note. I'm sorry, Professor, that I put my note on the chat. Sorry.
Okay. Yeah. So just move to the next question since Prof. Gelvin cannot access their chat. So I will read. Professor Gelvin, sorry if I deliver my questions into the chat box. First, I would like to express my gratitude for your presentation. In fact, it inspired me greatly because it contains newest and interesting information. If I may, I would like to confirm that as you noticed before, that VIR-D2 plays a role in Tdna integration. However, I am still unsure of the domain's role in Tdna integrations. Although VIR-D2 protein remain attached to Tdna, to Tdna strands as they enter the nucleus. VIR-D2's role in Tdna integration, if any, is unknown. VIR-D2 is involved in many processes upstream of Tdna integration, as far as I know, and mutation in VIR-D2 that impair integration can also impair one or more of these processes. So do they have this potential issue? Thank you for your kindly explanation from Suwasdi. Yeah.
So the answer is yes, of course. VIR-D2 is involved in many upstream processes, but that's why we look, when we're trying to block Tdna integration, we also look at transient transformation, which requires Tdna to get into the nucleus and become double-stranded. So if you get good transient transformation, by definition, the Tdna has gotten at least into the nucleus. So that's what we're trying to do, is unlink the process, let Tdna get to the nucleus and stop at that point. What is the role of VIR-D2 in integration? The exact molecular role, we don't know. Obviously, VIR-D2 has to be removed from Tdna prior to or during integration. There's a paper from the Tysterman laboratory recently showing that there's a protease that may take it off, or there's another way of clipping the DNA and takes off the VIR-D2 plus a couple of nucleotides. But how do we think VIR-D2 is involved in integration? We don't know. I could speculate. One possibility is VIR-D2 will target proteins at a double-stranded break site. Perhaps it will look at certain chromatin structures, or it will look at DNA repair proteins that have been attracted to the double-strand break site and target the Tdna to that region. Those are experiments that we would like to do, but we don't know, and this is just a hypothesis at this point. I would be very happy to hear somebody's suggestions about what the role of Tdna integration may be.
Thank you. Okay, Pak Suwasdi. The next question is from Dr. Sinto. Prof. Gelvin, I am amazed to be introduced to the idea that transformation that is not ended up with integration is useful in genome editing. In the study you have reported, the plasmid containing mutated VIR-D2 did not cause tumor, and so we can say that is not integrated. Is there any possibility that the Tdna was integrated but did not cause tumor, or it was not expressed? Regards to Sinto.
Yeah, so that there's always that possibility, although Tdna integration and silencing at this high level is unheard of. It occasionally silences. However, you're absolutely right. And we are actually checking this right now by molecular methods to show that the Tdna in these edited plants actually has not integrated. So we're checking that right now.
Thank you, Prof. Gelvin. Any other question from the audience?
May I, Prof. Darda?
Sure. Yeah.
Yes, thank you, Prof. Gelvin. Very nice presentation. So I have some questions. First, could we, if other virgin from the Agrobacterium only induced after the Agrobacterium contact with the plant cells, or can we induce?
Could you say that again, please? I didn't hear that. Say that again, please. Your question?
Yes, are the virgin from the Agrobacterium only induced after the Agrobacterium contact with the plant cells, or can we induce them before we use them for the transformation?
Yeah. Okay. So VIR-A and VIR-G are always on because those are the sensors. Okay. So they have to be making their proteins. However, after the bacteria contact these phenolic compounds, whether or not the phenolic compounds are coming from the plant, or you buy acetosyringone from Aldrich Chemical Company and put it in the medium and you induce the VIR genes, these VIR genes, the VIR-A and VIR-G are further induced, and all the other VIR genes that were uninduced now become induced. And the answer is yes, of course, you can induce these VIR genes on your own by putting them in the appropriate medium, low pH, low phosphate, and no sucrose, and you're adding phenolic compounds such as acetosyringone. So yes. Yeah.
So we can induce it in the sucrose medium.
No, not sucrose. No, don't use sucrose. Okay, use glucose. Sucrose is a non-inducing sugar. You have to have pH between 5 and 6. You have to have low phosphate, and there are various inducing sugars such as glucose. I think arabinose. There's a whole bunch of them, but sucrose is not an inducing sugar. It's not recognized by the CHVE protein. So you need the right pH, the right phosphate concentrations, and the correct, the co-inducing sugar, and then the phenolic inducer. Those are the important things.
Okay, thank you. May I one more, Prof. Darda?
Yeah.
Okay. How can we increase the cell susceptibility to be infected by the Agrobacterium?
I wish I knew that answer, because then we could transform anything. We do know cytokinins will increase susceptibility. Some Agrobacterium strains actually have a cytokinin biosynthesis gene in the VIR genes. I'm not talking about RPT. I'm not talking about in the Tdna, but they have a VIR gene called TZS, and TZS is involved in making a lot of cytokinins, and that does help transformation, but it's not essential because many Agrobacterium strains don't have that gene, and they still transform. One of the projects that I've just written a grant for and just submitted the grant just a few weeks ago is to try to understand why are some plants susceptible and some plants resistant. And it's not even that. Why are some very closely related varieties of plants? There's huge differences in susceptibility. So for example, many years ago, we looked at over 40 ecotypes of Arabidopsis. Some of them were extremely easily transformed, and some of them were virtually untransformable. They're all Arabidopsis thaliana, they're just different ecotypes. Same thing happens in other plants. We know that if you do crop plants, some genotypes of crop plants are pretty susceptible and some are almost impossible. The question is, why is that? Okay, and that's one of the things, that's the main point of the grant that we just submitted. We don't know, and I would really like to know that.
Okay,
Because
thank you.
once we know what causes susceptibility, then we can start manipulating that.
Thank you.
Nice. Thank you. There's one question in the chat from Dr. Yudiwanti, the head of a study program of plants breeding and biotechnology. Many thanks, Prof. Gelvin, for the very interesting presentation. For me, it is amazing. I'm sorry if my question is ancient. As far as I know, Agrobacterium tumefaciens naturally infected only dicotyl plants, but it can be used to transform genes into rice with this monocotyl. So, would you please explain the phenomena? Thank you very much. Regards, Yudiwanti Wahyu.
Okay, so when people first started looking at Agrobacterium, the phenotype they were looking at was crown gall tumors. So it happens on a very, very broad range of dicots, as you're correct. And it also happens on gymnosperms. You could make crown gall tumors on certain pine trees and things like that. Monocots don't get crown gall tumors, but you can transform them. They just won't get tumors. So people thought that you couldn't transform them because they were looking for the wrong phenotype. But if you use other types of phenotypes, such as, you know, making them resistant to an antibiotic or GFP expression or something like that, then you will see that under the right circumstances, they can be transformed. They just won't make tumors, probably because the hormone balance that causes tumors in dicots and gymnosperms is not right for monocots. So it's a very good question, actually. And it's just because of what people were using as a definition of transformation. They were using tumorigenesis as a definition of transformation, and monocots just don't get tumors. But that doesn't necessarily mean they can't be genetically transformed.
Thank you, Prof. Gelvin. Bu Yudi, answer your questions. Okay. Any question from the audience? Pak Awang, any questions?
Thank you, Pak. I think it's no from me. Yeah.
No, okay. From others? Boleh pakai bahasa Indonesia, ya nanti dibantu diterjemahkan.
One more for me, Prof. Darda.
Okay, Bu Dewi. Okay.
Yes, Prof. Gelvin, when I try to use the minimum medium to grow the Agrobacterium, we cannot get the enough concentration of the Agrobacterium for transformation. Could you explain this phenomena, or how can I get the enough concentration of the Agrobacterium when I use the minimum medium to grow them?
Well, it depends on what minimal medium you're using. I don't have enough information. Agrobacterium can utilize both sucrose. It could use either sucrose or glucose as a carbon source. You can grow it on either one. We use a minimal medium called AB minimal medium, but there are many different types of minimal medium that could be used. If you're, of course, the bacteria will grow much more slowly on minimal medium than they will on rich medium. When we're doing transformation assays, we will streak the bacteria out first on minimal medium to make sure it's Agrobacterium, because lots of things could grow on rich medium, but very few things can grow on minimal medium. So you could actually see Agrobacterium. They look very different from E. coli and other things. So we streak them first on minimal media to make sure that we have pure colonies. Then we just grow it in rich medium before, with the appropriate antibiotics for whatever your binary vector is. We'll grow it in rich medium before we do our infection assays. We do use minimal medium basically as a diagnostic to make sure that our strains, our cultures are pure.
Yes. Thank you, Prof. Gelvin.
Any questions from audience?
I have a question, Prof. Darda.
Please, Uyudi.
Yeah, thank you very much for Gelvin. It's very interesting presentation. The insert itu genome to the plant is randomly. Then interjected to the plant genome is random. So, is there any treatment to increase the probability it will insert into the desired location maybe? Yeah. Is that any? Thank you.
Well, there may be one way of doing it, and that is if you use CRISPR to make a cut site in a particular gene, that tends to be a Tdna trap. So you won't get all your molecules, all your transformants won't have Tdna there. But some people have said very high percentages of their transgenic plants after CRISPR editing sometimes have Tdna, in the right there, because you're making a double-stranded break. Double-stranded breaks tend to be Tdna traps. So it's not going to be 100%, it may not even be 10%, but maybe a few percent of them will be at that region, at that particular locus. But you will also get Tdnas elsewhere also.
Thank you very much.
Thank you, Yudi. Thank you, Gelvin. There's one question from Aditya in the chat. Thanks for the presentation. I would like to ask if there a difference in the methods of transformation used in dicot and monocot plants induction. I see difference in the CRISPR type in the form of Cas9 monocot and Cas9 in dicot from Aditya and Subchang.
So can you repeat the very first part of that question again?
Okay. Is there a difference in the methods of transformation used in dicot and monocot?
Okay, so basically people, from what I understand, people work out what is best in their laboratory for a particular species or even a particular variety. So I don't think there's any sort of one size fits all that we always do things in the same way. A lot of it has to do with the tissue culture of the plant. So people generally use transformation protocols that match the tissue culture regime of the plant. So there's no one way to transform a plant that can work best with dicots or monocots. People match it to their tissue culture conditions. So,
what's regime that you use in transformation? What's the protocols of?
Well, when we're doing a Arabidopsis or tobacco, it's so easy to transform a Arabidopsis or tobacco that you could do almost anything that it would work. Arabidopsis, we use cut root segments because we're a basic science lab and roots are actually the natural target for Agrobacterium. But tobacco, we use leaves, infiltration or leaf sections and things like that. And the Arabidopsis, of course, you could do flower dip, which is a very unnatural way of doing things, but that only works for Arabidopsis pretty much in a few very closely related species. So people basically work out what is best for your species and your variety. It's a combination of what's going to transform well, but also what will regenerate plants well. So you have to keep in mind the regeneration protocol. And that's why people are using these new developmental genes, the Wuschel and baby boom and things like that, because they stimulate regeneration. And so that's starting to become very popular now, mostly for grasses, monocots. It's only beginning to be used in dicots now. It doesn't work as well in dicots from what I understand. But it works well in some species of monocots. But that's not transformation. It's not getting Tdna in. It's recovering a regenerated plant. So there's two parts to transformation. There's the getting the DNA in part, and then there's getting a plant back out of the tissue part. And you have to be able to match both of those things.
Okay, thank you. Maybe one question from me. That's how's the future of the Agrobacterium in the era of transgenic, and then genome editing, and then also some people try to use nanotechnology to deliver the DNA cargo from outside to cytoplasm. So will be the future of Agrobacterium is still bright or what?
Well, I hope it's bright, at least until I retire. No, no. So nanoparticles could be used for a lot of different things, but people haven't completely worked this out yet. And also the chemistry and the delivery systems are not, are highly specialized, and they may not be obtainable in the average laboratory. Agrobacterium is very nice because you don't need specialized equipment. You don't need specialized synthesis things. You don't need specialized delivery systems. Agrobacterium is just naturally doing it. So people tend to go to Agrobacterium when they can. When Agrobacterium doesn't work well, that's when they start going to guns and nanoparticles and things like that. The first question you asked actually is something which is very particularly interesting to me. And if you just said, what is the future of Agrobacterium, and just stopped right there, okay? And for me, that's really important. And the reason is that there are virtually no labs left in the whole world that do transformation research. People do work on Agrobacterium, but looking at genome evolution of Agrobacterium plasmids. They're looking at the pathology of Agrobacterium. They're looking at things like Agrobacterium cell division, a lot of microbiology and things like that. These are all very, very important things to do. But there's almost no laboratories left in the whole world right now that work on the mechanism of transformation. Almost all of my peers have retired, virtually every one of them. And my mentors, that generation, they've retired or died. And so we really need young people to get into this field because the field is sort of collapsing, basically. So I encourage young people to think about careers in basic agrobacterium science. And people say, oh, we know everything we need to know about agrobacterium. We just give me your strain and tell me how to transform a plant. We certainly don't know a lot of things. And it's the things that we don't know that are preventing us from transforming a lot of plant species or from improving agrobacterium as a tool. So I'm a very firm believer in doing basic biology, basic science, letting the basic science guide us into the more applied types of things. I'm not against applications. I love applications too. But my true love is the basic science. And it's really important for young people to get into this field so that the field doesn't die, basically. I feel very lonely right now. I'm 73 years old, and virtually all of my colleagues have retired. There's nobody working on this anymore. So we need people.
Sure. Yeah, we hope some new generation will come working on Agrobacterium. Basic science or in basic science or in transformation for commercial.
One of the ways you can do this, by the way, is to have collaborations where people send their students to laboratories doing Agrobacterium, the few laboratories left, to be trained for maybe a year or so, and then they can go back and start their own groups. If the groups don't want to work on their own, start from scratch, start it as a collaboration. Come to some laboratories, a few labs that are left, learn the system, and then go back and start your own labs.
Yeah, we will try. Okay, there's more question from Dr. Sinto. I will read. Prof. Gelvin, one of the challenges in plant transformation is the Kale regeneration. I remember in one part of your lecture, you mentioned about the component important for the Kale generation, callus or Kali regeneration. I'm sorry that I forgot about it. Was in the FIRD2 or in the DNA repair related genes?
So what we found was in rice, a DNA polymerase theta mutant was deficient in regeneration from callus to plants. So if you didn't have a good DNA polymerase theta gene, then you couldn't regenerate. And we don't understand why a mutation in a DNA polymerase should have an effect on regeneration. It's something that actually that I've encouraged my Japanese collaborators, Ayako Nishizawa Yokai, to look into. In terms of other aspects of regeneration, that's one of the problems with agrobacterium-mediated transformation to get a transgenic plant. You could get the DNA in, but sometimes it's hard to regenerate a plant. People have been using these developmental genes more in the last 5 years or so, the baby boom, Wushel, and various other types of genes to try to do regeneration. And they've had a lot of success in many cases. There's also a new move in the last few years to try to do what's called implanted transformation, where you transform a plant tissue right on the plant and try to regenerate a plantlet right out of the transformed tissue. There's some people who have been working on that, so it bypasses the tissue culture step. And the tissue culture step is one of the most difficult steps. First of all, it's very time consuming, very expensive, and requires lots of plates and lots of lab space and things like that. And the other problem with tissue culture, of course, is that in many species, when you put plants into tissue culture and regenerate them, you get somoclonal variants. So there's a lot of mutation that goes on in tissue culture during regeneration. Transposons jump around, various other things happen in tissue culture. It would be nice if we could completely bypass the tissue culture phase, make things faster, much less expensive, and much easier to do if you could do that. So Dan Voitis's lab in Minnesota has been one of the key labs in trying to do this and trying to get plants to regenerate directly out of, for example, infected leaves of tobacco or tomato. And this is another way of going. And all my tissue culture friends tell me this is the way it's going to have to go. If we're going to get massive amounts of transformation in labs that don't have a lot of resources, you have to try to bypass tissue culture in some way.
Okay. Thank you, Prof. Gelvin. I hope Sinto can, okay. And then we have new question here from Zuyasna. Thank you for the nice presentation, Prof. Gelvin. I have a question. What about transformation using biolistic gene gun? Which one is the best compared to the Agrobacterium transformation to get success transformation? Thank you.
Both methods are fine, but each one has its own problems. If you talk to Freddie Alpeter, for example, he's going to use gene guns all the time. Guns bypass the problem of host range of Agrobacterium, but guns have their own problems. You tend to splatter up the genome with multiple copies of fragmented DNA all over the place. It's much more difficult to control integration and copy number and keeping the DNA intact using a gun. So yes, it could bypass the host range problems that Agrobacterium has, but then you have other problems of, the resulting transformants are sometimes a mess. Now, another thing that people can do is not using DNA as such, but using ribonucleoprotein complexes of CRISPR with the of CRISPR complexes, the Cas9 protein with the guide RNA, and you put these nucleic acid or ribonucleoprotein complexes into the cell, they edit, and then they just disappear because there's no DNA there. And this is a method that people are looking at. The problem is delivery. How do you get these ribonucleic complexes into ribonucleoprotein complexes into a cell? You could do this in protoplasts, of course. But then there's a problem of regenerating protoplasts back to a plant, which can be done with some plants and more difficult with other plants. So these all indicate that it's not just, the CRISPR technology is fairly advanced now. Of course, it's going to advance much more, but it's fairly advanced. We know how to make lots of mutations. We know how to do base editing. We know how to do all sorts of things. The problem is delivery the system. How do you get these CRISPR reagents into the cell and to be able to regenerate a plant back from them? So this is why I think this is why we started working on our VRD2 work, because we know how Agrobacterium works. If we could only prevent Tdna from integrating, but still getting it, the genes to express, then we could start doing a lot of things with agro. That's one of the things that guided us in our initial experiments.
Okay, thank you, Prof. Gelvin. One more question, I think, from Buari. I'm sorry, I am a beginner in this topic. How do disarm a pathogenic Agrobacterium tumor patients to be factor for genetic transformation so we could have our own isolates, no need to buy anymore? It is really genetic modification or just a certain treatment? So how to disarm? Yeah.
So you need to, what we call disarm the Tdna, which means you have to delete the oncogenes. By the way, you don't necessarily have to buy things. Sometimes labs will give it to you. The problem is customs, though. Getting bacterial strains through customs is sometimes a difficult process. You have to be able to delete the oncogenes from within the Tdna. That requires some rather sophisticated microbial genetics. But just recently, Kan Wang published a paper in which they're using transposase, CRISPR-mediated transposase-guided deletion technologies. And this could be used to delete genes within the Tdna. So previously, the microbial genetics was really complex and difficult to do in a lab that doesn't normally do this sort of work. There's no chemical that you could just add to Agrobacterium that'll automatically disarm it. You just kill the Agrobacterium, basically. But there are genetic tools that could be used. So you could, for example, isolate virulent Agrobacterium strains from a plant that it infects really well and disarm it, perhaps, so that it works well on that plant without making a tumor. So there are microbial genetic tools to do this.
Okay, thank you, Prof. Gelvin. I think maybe the last question from me, if any possibility that's part of sequence of others than the Tdna that's transferred from the Agrobacterium into the cytoplasm?
The answer is yes. We do know that all sorts of DNA from Agrobacterium can get into the plant, can actually integrate along with Tdna. So we were the group that first showed that binary vector sequences from the backbone of the binary vector could get in. But other groups and our group also have now shown that all sorts of different sequences from Agrobacterium can get into the plant. We don't know how. From the Agrobacterium chromosome, from other plasmids that happen to be in Agrobacterium. We know that these DNA can get in and it can integrate, and this can become a regulatory problem because the regulators want a clean insert. They don't want all this other stuff in there. Another thing that happens that most people don't appreciate is that sometimes Tdna sort of gets into the plant and then it sort of blows up into little bits and pieces that integrate. And if you're doing standard PCRs or DNA blots, it's really easy to miss this because you don't know what to look for. The only way you're going to catch this is by whole genome sequencing. And then all of a sudden you find these little bits and pieces all over the place. The regulators certainly don't like that either. So whole genome sequencing is becoming essentially essential if you're going to do field release of crops. Just doing some PCRs and DNA blots is not good enough.
Yeah, thank you. Because some pro and contra of GMO still talking about the DNA from the Agrobacterium that's go to the cytoplasm and
So that's why if we could get Tdna to get to the nucleus but not integrate, but the CRISPR reagents encoded on the Tdna to do their work, and then everything just disappears. There's no transgenic plant. There's only an edited plant at that point. And that's what we're going for, the goal that we're going for.
Okay. Thank you, Prof. Gelvin, for the presentation and discussions. Very interesting. But our time is up. And I think that the more I learn, the more I feel that I know nothing. it's very interesting, very up-to-date about the Agrobacterium and genetic engineering and genome editing. So I give the time back to Ibu Dewi.
I just want to thank people. Thank you for inviting me to give this talk. I enjoyed doing it. I hope I stimulated a few of you people to consider research careers studying Agrobacterium. That would make me very happy if we could do that. But thank you for the opportunity to give this talk. And I hope someday to be able to actually visit you in person in Indonesia.
Oh,
Sure.
yes.
You're very welcome.
We hope very welcome, Prof. Gelvin. Could you stop your
Screen sharing?
Yes, okay, thank you very much. Yes, it's very nice time with you. 2 hours is feels so fast, so short. We still like to discuss more, but however, maybe you already almost midnight there. Already
No,
10.
it's only 10. It's still early.
But yes, maybe we can get other time next time maybe to discuss more with you. Thank you very much.
I'd like that.
Yes, thank you for all participants that already give questions and comments. Thank you for joining us. Prof. Gelvin, we cannot reply your time. We can only give you a certificate.
Okay, that's fine. That's all I need.
Certificate, could you? So this from our department head.
Oh, thank you.
Thank you very much.
Could you email that to me?
Yes, I will email
it
to you. Thank you very much. So maybe we can stop now, or maybe we can make a group picture.
Yeah.
Okay.
Just open the camera.
Yeah, please, all audience, open your camera. We will get a group picture with Prof. Gelvin. Okay.
You have about 150 people there, right?
Yeah.
Big group.
Some sleep already.
Big group, yes. Everyone can take picture. Me too. We have Professor Sudarsono, Prof. Gelvin, Professor Sudarsono join us. He is our head division on biotechnology.
Very nice. Yes.
Yes. Professor Sudarsono, maybe you like to greet Prof. Gelvin, please, Prof. Dar, while I take a picture. Prof. Dar?
Prof. Darsono wrote in the chat. Thank you, Prof. Gelvin, for such interesting lecture.
Okay.
Terima kasih, Bu Dewi. So,
okay. Maybe he is on the way to the other place. Thank you for everyone. I already.
Should I leave at this point?
Sure.
Sure.
Sure. Okay.
Thank you again.
Thank you.
Thank you, Prof. Gelvin.
See you again, Prof.
See you
Gelvin.
around.
Bye-bye.
Bye.
Terima kasih Bapak Ibu atas kehadirannya, Ibu Yudi, Pak Awang.
Terima kasih.